All right, these notes go into your PBS course file. All right, these notes go along with PBS 115, where we're talking about genetics and genes and DNA. And so an important component is to understand chromosome structures. So a lot of times when you see chromosomes, they're just these little X diagrams. And so this is kind of representing that little X diagram. But in reality, this structure is a chromosome composed of two sister chromatids. So this is a chromosome and this is a chromosome, and they are bound together at the centromere to create a chromosome with two chromatids. The longer arm of a chromosome is considered the P arm. The shorter arm is the Q arm. This chromatid has a strand of DNA that is wound up on it. So that's what all these little circular diagrams are supposed to be. That's the DNA that is wound up, that is compacted, that is organized on that chromatid. And so an area, a region of a chromatid represents a locus. So this right here is the locus of a particular gene. And this is the locus of another gene. And this is a locus of another gene. If I were to unwind that strand of DNA, I would have my chromatin, which is the strand of DNA, and each section of it would represent a different gene. Okay. So this is a sister chromatid, and this is a sister chromatid. So this is a chromosome that has two sister chromatids. If I unwind that DNA, if I take that DNA that's wound up and I stretch it out, that would be chromatin. So chromatin is the strand of DNA. And each region on it represents a certain amount of DNA that codes for a gene. So for example, from here to here, all of that DNA codes for gene number 1. And this DNA codes for gene number 2, and this DNA codes for gene number 3. So a chromosome is the structure. A chromatid is a duplicate or a copy. This is a chromatid and this is a chromatid. So this is a copy of that. They're exactly the same. Chromatin is the DNA strand. So this is the DNA strand, which is chromatin. We'll be looking at two different types of cells, prokaryotic cells and eukaryotic cells. And so you should have an insert that gives you a diagram with each one of those. So I have not taped that completely down because I put information underneath. A prokaryotic cell is a cell that lacks a membrane-enclosed nucleus and membrane-enclosed organelles. So prokaryotes, no nucleus. But technically, it's cells that lack membrane-enclosed nucleus and cells that lack membrane-enclosed organelles. So they don't have membrane-bound nucleus or organelles. Eukaryotes do have a nucleus. Eukaryotic cells are cells with organelles and a nucleus that are enclosed in membranes. Eukaryote is a cell with an organelle and nucleus that's enclosed in membranes. Organelles are membrane-bound structures within cells that each have specialized functions. Organelles are membrane-bound structures within cells that each have specialized functions. I like to think of organelles within the cell. The cell is kind of the city, and the organelles are all the different little things that have to happen, the different jobs, the different stores and stuff that have to be part of that cell in order for it to survive. So organelles are things like the nucleus, or endoplasmic reticulum, or ribosomes, or lysosomes, something along those lines. All right, now on the next page. So DNA stands for deoxyribonucleic acid. So DNA stands for deoxyribonucleic acid. DNA are instructions of our cells. They are the information that makes us who we are. DNA is built by adding nucleotides to the 3' end. And that's going to make a little bit more sense in AP biology. But it's important to know that DNA is constructed from 5' to 3'. So DNA is built by adding nucleotides to the 3' end. Chromatin is the DNA fiber. Chromatin is the nucleic acid strand. I'm trying to accentuate the ends so that we've got nucleic acid strand, chromatin. That's the difference between those terms. So I'm trying to help use that. It is the strand of nucleotides that are bound together, the ATCG nucleotide strand. Chromatin is the DNA strand. A chromosome is the structure that the chromatin is wound up on or organized upon. So a chromosome is the structure the chromatin is wound / organized upon. Chromosome is the structure the chromatin is wound / organized upon. Chromatid is a copy of DNA or a duplicate. Chromatid is a duplicate, a copy of DNA. We're not focusing. There we go. Chromatid is a duplicate or a copy of DNA. We'll get genome here in just a second. So a gene is a section of DNA / chromatin. A gene is a section of DNA / chromatin. So when we unwind that chromatin strand, each section of it represents a certain amount of DNA, which represents a gene. And I had to put this on a sticky note, so because I ran out of room, but it needs to go in the same general area. So the genome is all of the DNA of an organism. Everyone's unique genome is their DNA profile. So a genome is all of the DNA in an organism. Your unique genome is your DNA profile. All right, so DNA has proteins. So histones are the proteins that are a part of the chromosome structure. The DNA is wound around the histones. So within a chromosome, you have that chromatin, that DNA, and it's wound up. Histones are proteins that the DNA is wound around. A nucleotide is a monomer of the DNA polymer. A nucleotide is a building block of DNA. So adenine, thymine, cytosine, guanine, those are nucleotides. Those are monomers of the DNA polymer. Just do the rest of this. All right. So DNA profiling. DNA profiling is a forensic technique that compares DNA sequences. Because our genome is all unique to us, we can use it for DNA profiling. So DNA profiling is a forensic technique that compares DNA sequences. So why would we want to DNA profile anybody? So these are some possible reasons: to match the suspect in crime scene DNA, for paternity reasons, for genealogy, for missing persons / body parts. So if you find a missing person's skeletal remains, or if you find part of them, then you can use that DNA to give a family closure, to find out what happened to their loved one. We can use DNA profiling to diagnose disease. We can use it for genetic counseling / gene therapy. And we can use it for biohistory, which is like anthropology. So lots of reasons why we would want to use DNA profiling. All right, so I'm going to take my genome, post it, and stick it right back over there by gene. All right, on the next 2 pages of your comp book, you will be going through 115, and you should have a couple of inserts, a couple of pictures with the DNA model that you're working on. These pictures should be labeled. They shouldn't be blank like they are now. Take some notes over what are these structures, what's going on. You should also have an insert about Watson and Crick and their information that they used in order to find Chargraph's rules. So make sure you read over this information because it's important. Then you should also be taking notes on 115 and 116 based on the PCR process. What does PCR stand for? What does it do? Where are sources for DNA for extraction? Where do we get DNA from to run through for DNA profiling? What are the steps of DNA extraction? Chances are you might have even done a DNA extraction lab in one of your other biology courses. And so those notes should be next to the pictures from that page. So this should be from 115 and 116 in PBS.